Okay, so welcome to this hangout session for the course Human Molecular Genetics. I hope all of you have found this course useful and easy to follow. To test how good you are with the subject and your understanding, we have been, sending you the assignments. I'm sure all of you are finding it easier and and useful as well. So what we will do is at first we will take up few of the questions that are already posted in this spreadsheet. The link was shared by the TAs. And in the meantime, if you have more queries, you could probably post them using the chat box that you could find on your screen. So let's look at some of the questions. Um, some of you have said this is good and so on. Thank you. Um, and there was one question by Ilya Mahedi, that is, why repeat nucleotide sequence are recurrent hotspots for mutations and what makes them unique in reference to other coding sequence. Um, I need to split this question into two. The first half, that why the repeat sequence are hotspots, that let me take that first, and then try to answer the second part later. When we talk about repeat sequences as hotspots, I think there are two different um, types of mutations in which the repeat sequence become hotspots. One is the recurrent deletions. There are deletions that happen within defined breakpoints, and this happens because of a large segment of the DNA are repeated, meaning the same sequence is present with an interval of some unique sequence. Therefore, during the meiotic recombination, these segments undergo what is called as unequal recombinations, a topic that we discussed when we were discussing about the microsatellite markers, as to how the new alleles come. So when such kind of unequal recombination takes place, the regions between the two repeat segment are either deleted or they get duplicated, and you end up having one or the other disorder, depending on whether the disorder is because of the loss of function when a given segment is deleted, or you have three copies of the segment. If it has got any gene, then that gene number also becomes three in number. As a result, you may have trisomic condition for a small segment of the DNA, like what you see in Down syndrome and so on. So that's the reason why the repeat elements can cause this kind of recurrent mutation. The other type of mutation that are associated with repeats are the what we call as dynamic mutations, wherein the repeat undergo massive expansion during meiosis, where it is because of the repeats attaining certain structures. As a result, the DNA repeat number increases. That's for the first part. The second part is what makes them unique in reference to other coding sequence. I guess you are referring to the trinucleated repeat sequences. That's where the repeats are present in coding sequence. Most of the other repeats are not present in coding sequence. So a simple reason is that if the repeats are not multiple of 3, any increase or decrease in the repeat unit would affect the coding sequence. Therefore, it would affect the DNA coding potential. Therefore, only the triplet repeats are allowed in the coding sequence to undergo expansion or contraction. And again, whether they are selected depends on whether the protein having such amino acid repeats have normal function, or it affects its function, and so on. So repeats in general are rare in coding sequence. Is more common in non-coding sequence, including the promoters or UTRs, and more so in regions where there are no genes. So that's the answer for the second part of the question. There was a question from Dr. Saha regarding lab visits to learn more practical ideas. That's a good suggestion. What I would recommend is that since there are every year this course is being offered and other courses are being offered, there are a large number of students who credit these courses, so it would be practically difficult for us to host all the students or great majority of the students who are interested in having a practical. We do, however, accommodate requests from a few students, maybe two or three in a given summer program. So that is called a surge program that IITK offers. That is mainly for those students who are in BSc, if you come through the Science Academy, or if you are an MSc students or BTech students, you are in the final year or fourth year, then you may apply, using the surge program that is available. And of course there is a it's a paid program in the sense that the institute supports, and of course you look at the merit list as well. So that is something that I would recommend. You can, those who are interested may apply. It is not only my lab, there are large number of labs here in this department that have interest in molecular biology, molecular genetics, human genetics. One or the other may find you suitable. Then you are encouraged to apply. um, there was a question, I still don't get exactly what the question is, how to explain that molecular genetics present in our human body. I don't really get what I meant, what possibly the meaning of the question. If Vingeshwarar is around, if he can post it again using the chat box, it would help me to understand your question. I would certainly like to answer. Dr. Deshmukh, a pediatrician, is with us, and um, he posted a question that he needs to have more basic discussion on pattern of inheritance, autosomal dominant recessive X-linked or Y-linked disorder. Um, we could discuss, certainly. It all depends on the family. What we have given in this course is a more of a hypothetical family, which sort of makes the inheritance more obvious. But it's not necessary that every family that shows a inheritance pattern for a phenotype that has a genetic origin to be as explicit and easier for us to judge whether it is dominant, recessive, X or Y. It all depends on how many members in a family are there, and how informative the family is, whether it is multi-generation, the sense that three generations and so on. But what normally done in clinical practice, if you are interested in that, is that you basically look at the clinical features and then you sort of map the clinical features to a given disorder and look at the literature as to what has been told about this particular clinical feature, whether it has a genetic origin. If so, what kind of a mode of inheritance people have seen in the families that they have investigated. With that, you try to recruit more and more families and then try to judge as to what could possibly be the mode of inheritance. So it would be very, very difficult to arrive at any a kind of a conclusion with regard to the inheritance of a phenotype if you are looking at only one family. I would say that is risky, not advisable. One has to look at a large number of families, and these families, you sort of uh, set up a stringent criteria for the inclusion exclusion. Therefore the disease phenotype is as homogeneous. Therefore you are removing the syndromic cases which may have origin elsewhere, which may have a different genetic basis, and then try to understand. So this is for disorders that have unknown etiology. You don't know where the gene is located, what kind of mutation they're in. But for disorders for which the gene has been identified, I would recommend you to go to a database called OMIM, OMIM. This is online Mendelian Inheritance Men. That is a database. If you give the query sequence for the disorder, you'll be able to get all the details and the literatures supporting that kind of inheritance. That would be very, very useful to you. Um, there's one, I guess, Miss Lata. She is a faculty. She has a question as to what are the legal and ethical implications of Indian Human Genome Project. I don't think that there are any kind of ethical implications that are unique to Indian Genome Project. Whatever that the guidelines that have been given for the International Consortium for the Human Genome Project is pretty much is being used, except that if you are an investigator who would like to um, recruit individuals for isolating the DNA for characterizing the variation that could be unique to each of the groups, then you would like to have a kind of guidelines that are put forward. You can look at that there is the Indian Genome Consortium, so that is led by the institute IGIB in Delhi. They have published a paper. Later, you may want to write to us in the discussion portal. We'll share with you the publications, which clearly define as to how you define, for example, the Indian population, and how you have to select the individuals from different groups, and what are the important aspects that you have to consider if you are starting human genome related research in India. So that's the only with reference to the sampling and how you are comparing, for example, the cases and controls, and what would be your controls, where the control should come from. These are the guidelines that are given, but otherwise, there is nothing really that is very different from what has been recommended for the International Human Genome Project. Legal aspects, and of course, most of these studies, the identity of the individual is not revealed, and their liabilities and other things are protected. You cannot use it for any of the legal cases. These are anyway, these are as per the guidelines that is given by the ICMR. You can go through that as well. This is not only for the Human Genome Project. For any of the human genetics project, if you are involving human subjects, then these are sort of guidelines that you use. You pretty much use a similar kind of a guidelines. Um, okay, so now let's look at there are questions. Legal implications I have just now explained. Um, there was a question about the pattern of inheritance. I just sort of given you some overview on that, and there was another question. I have been watching your wonderful videos. Unfortunately, I missed to register for the exam. I request you to kindly inform me if I can still register. I guess that is not in our hand. As I understand, the last date for registering the exam is already over. But I would recommend that if you are interested in the topic, you may continue to watch the videos. This course will be again offered sometime next year. You can register again, and you can get the credits. Um, Give some more book or link for the knockout and pedigree. Sure, we would like to add. What we are doing right now is the enough reading material. We are not loading you with more. But if you are interested in learning more, do write to us. We will add, especially those who are interested in studying, we will separately share more link for them. Exact mechanism in all the pattern of inheritance. Um, I still don't get the question. What do you mean by exact mechanism? So basically, there are two major um, what two major types of inheritance. One is what you call as a dominant. Other one is a recessive. Um, you this is the major thing, and then there are a large number of differences between these two. These are discussed in the handouts as well. We would share with you some more links on that, and then you need to practice it. You have to take as many pedigrees and then try to practice whether they follow these guidelines and whether you can do. So the main difference, of course, is recessive. You will not see it in every generation. Dominant, you will see it almost in every generation. Affected parents, one of them would be affected because one allele that causes the disease, and then it could be the affected could be a male or female, can be transmitted by a male or female, and so on. So that's where it tells you whether it is dominant and it is autosomal, and that's the major differentiator between the recessive and the dominant. Um, uh, so that's about the inheritance. Um, could you explain more about loss of function, gain of function? It's a simple concept when you say loss of function. So that phenotype results because the gene has lost its function. That means either the gene is not expressed or it is expressed but the protein is not functional. So in result that it is equivalent to not having the gene. That is called as loss of function. A gain of function is that the gene is there, there is a mutation as a result. Either the RNA or the protein acquires some new function, and as a result, you end up having certain disorder. One classic example is that probably is discussed in the class as well is androgen receptor. It's a protein and which functions as a transcription factor. So if you have mutation that affects the function of the androgen receptor, so when there is androgen, when it binds to the receptor, this receptor is, if it is having a loss of function mutation, the body would assume that there are no androgen in the system, therefore the body should develop towards a female. And there are conditions wherein individuals that are XY will develop as phenotypically female, um, and that is because of the loss of function. So it is, the mutation affects the androgen receptor's function. As a result, androgen is ineffective. But the same protein, if it gets a different kind of a mutation, for example, the repeats that are present, polyglutamine, if it expands, it doesn't affect the androgen receptor function. It's still that individual, if he is XY and androgen is produced, then individual becomes a male. But the repeats, if it expands, then it causes this androgen receptor protein to form aggregates and then so become toxic to the neurons, and the neurons slowly die. As a result, these individuals would develop a neurodegenerative condition, and that's because of a gain of function. So you have, because of the expansion of the repeat, the protein acquired a new function, and and therefore you have the disease. This is the definition. Clearly, loss meaning the function of the protein or gene is lost. Gain is that it has acquired a new function. So that's what it is. It could be at the level of protein. For example, a mutation um, in an enzyme may lead to the enzyme identifying a new protein as a substrate. It could be another example. So there are many such examples, but essentially it is a new function that it gains. There's another question from Dr. Okay, Dinesh Kumar. So that's already Murali Krishna has answered. Uh, can you prefer any reference book? Certainly, there is a reference book, but this book is not available free online. Therefore, we will not be able to recommend online. But if you are interested, we could share the book. It is the book name is Human Molecular Genetics by Strachan. That's what it is. You can search, and in case if you are unable to find, you send us, email us. We will refer to you that book in Amazon. If you are interested, you can buy that. Um, so there's another question: is it possible for you to start some short fellowship distance learning, especially for pediatrician? Um, I mean, we could, but what I would recommend is that there is a far more structured program being run by IGIB, Institute of Genomics and Integrative Biology at Delhi. There is Dr. Sridhar and Dr. Vinod Skaria. They run a much larger program for rare disorders, especially that affect the pediatricians, and they have a structured program, a network involving a large number of pediatricians. And maybe if you're interested in, you can get in touch with them because it is a consortium model wherein they involve a large number of pediatricians for the genetic screens. So I would recommend you get in touch with them. So therefore you can even get in touch with in the medical or clinical geneticists to work in diagnosis and genetic characterization. So that would be the best model that I would recommend. Um, there's another question for DNA breakage and translocation. Um, it's again, these are, when I talk about translocations, this happens because of the double strand break, because the entire segment of the part of the chromosome breaks and, gets translocated elsewhere. And in some conditions, it is a recurrent. For example, in the one of the cancer type, it is a recurrent, meaning that, exactly at the same position, the chromosome translocations happens, and often people believe that it could is because of certain structural elements that are present in these chromosomes that are making it more prone for such deletions and translocations. Other than that, we don't really know what is the mechanism by which it happens. Um, there is another question: conventional approach to position cloning. How to identify the candidate chromosomal region? Um, the candidate chromosomal region is called as the positional cloning, in the sense you have used a large number of markers. So these markers are present across all the 23 chromosomes, pairs of chromosomes, and these markers are nothing but microsatellites, each one having different alleles. You basically take a couple of families, run through these markers, and see which allele of which marker more often co-segregate with the disease phenotype, and whether that marker, co-segregating with the disease phenotype is common in more than one family but having the same disease. That indicates that that's the region of the chromosome, or that chromosome is involved in the disease, because if there is a gene that is mutated causing that disease, if there is a marker present next to it, and that allele would certainly be co-segregating with the family. So that's how you identify a small segment of a chromosome to be, the region that could potentially have your candidate gene. Can you give some more examples of pedigree in video? It would be difficult to show a pedigree in video. What we could do is that in the portal we'll add a few more exercise. Okay, we will give you a PDF file we upload with certain some worked out ones, some for you to test it out. We'll add it next week. That would be better. Kindly explain calculation methods for probability in Mendelian genetics with normal, monohybrid, dihybrid as well deviation dihybrid ratio. When we talk about the human genetics, we really don't say it is hybrids or monohybrid, dihybrid, because you don't set up any crosses. We basically infer from the crosses that has happened. For example, the marriages. So you really don't select phenotype either to set up. When you talk about monohybrid or dihybrid, you are taking contrasting phenotypes. For example, the Mendelian classic example is the height, the height of the plant, or the shape of the seed, or the color. So these are contrasting characters you take up and set up crosses. These are all, simply cannot be done in humans. So basically, you look at phenotype that are having certain disease, very contrasting phenotype that runs in the family, and with the kind of inheritance of that, you try to infer whether it is Mendelian, in the sense that caused by a single gene defect, and whether it is autosomal, recessive, dominant, and so on. And dihybrid and all, it is impossible, because how are you going to get, I mean, this is like, you forcibly marrying two different individuals having two different disorders, and looking at next generation, how many of them have two disorders, and so on. These are not, something that you do it in human genetics. So that's something that is more for the experimental model. Is there any resource to check our understanding regarding pedigree and other concepts covered in your lectures? Um, um, well, I don't think that there are kind of an online module wherein you can sort of try and answer and see whether what you are saying is correct, to the best of my knowledge. But what we will do is we'll try to add few more exercise in the portal, so you can take home and then do it on your own. Um, does lot score provide exact count of markers? No, lot score doesn't give exact counts of markers. It is the markers that help us to arrive at a lot score to tell whether the co-segregation of a given marker is by chance or random probability, or because there is a gene that is present very close to the marker, therefore it is co-segregated more often. So it is basically you look at the probability whether it happens by chance or it happens because there is a gene present next to that marker. So if higher the lot score gives a confidence that there is a gene present next to that marker, therefore it co-segregates. Um, um, can you throw some light on what is that? On factor maintaining chromosomal integrity. I don't think that we have really understood the chromosomal integrity. I guess you mean the way by which chromosome remains intact, not mutated. I guess that's what you are meaning. Um, there are, there are dedicated machinery that takes care of the errors within the DNA when the DNA replicates. That is one of the first set. And of course, after that, you have the higher order structure of the chromosome, where you have histones and other proteins that keep the DNA protected. It's not a naked DNA. It's always covered by the protein. That's probably one of the other reasons why it is kept intact. The third is that the chromosomes within the nucleus, each of the chromosomes have a territory. There are marked regions within which the each chromosomes remain. That's what we understand now. There are ways by which the cell is able to anchor each DNA in different segments of the nucleus. And perhaps these are some of the mechanism that the nature has evolved to sort of maintain the DNA intact. But certainly we don't know all the mechanism, right? Um, yes, sure. We will post it on the anything that we do. We'll post it on the course portals. Therefore, you can we can all of you can get benefited. Can you explain a consensus coding? I'm not too sure that what you are trying to say. Maybe you can rephrase your question and post it again so I can answer it. Um, will you repeat the name of the institute? It is IGIB. It is one of the CSIR Institute for IGIB. Institute for Genomics and Integrative Biology. It's in Delhi, so you can search. You can just simply give IGIB. You will be able to get. It's simply give CSAR IGIB, so you'll be able to get that sequence right. Yeah. It was mentioned that a great majority of the recessive disorders are caused by loss of function. Dominance caused by gain of function. How do we know that? Okay, it's because for these two groups of disorders, people have identified the DNA, and then after that you have sequenced the DNA, and then try to understand how the variations in the DNA may affect the gene function. So more often for recessive disorders, it's the mutations are like a nonsense mutation, meaning in a place where you have a codon for amino acid, it's now stop codon, or there is a frame shift mutation, or there's a deletion of the entire gene or part of the gene and so on, which would invariably affect the either the transcript being made, or if the transcript being made affect the protein being made, or even if the protein is made, the protein is non-functional. That's why it is loss of function, and both copies are gone. Therefore, because heterozygous are normal because the one copy is good enough to take care of the normal function. But in a gain of function disorders, like for example dominant disorders, just mutation in one gene is able to manifest the disease condition. That means more often this mutant allele is dominant over the wild type. The wild type one copy is not good enough. The example being, the triplet repeat disorder that we have seen. A repeat expands and the repeat bearing protein becomes toxic, and the toxicity is a new function that is conferred by the mutation. So therefore, in majority of them, it is a gain of function. But there are exceptions we have discussed. These are some dominant disorders are caused by loss of function mutation, but there it is called as what do you call as haploinsufficiency, meaning one defective allele.
Excuse me. Live session cycle.
One recessive allele affects, um, you know, the is not good enough for the cell to carry out the function because half of the protein is not good enough. But there are exceptions, but majority of them are, what do you call as gain of function mutations. Dr. Abhishek, please throw some light on the CRISPR-Cas. Yes, I mean, this is a new field that is coming up where we will be able to engineer the DNA using set of enzymes. Whatever kind of a mutation that you want to create, you can create to create models. Or now there are some um, studies which suggest that you can fix the mutation and reverse that to wild type, and there are some claims from some country that you can correct even the genetic disorders, but still there is a controversy. But I have another course called functional genomics. The again this videos are available in the YouTube, and where we have discussed this CRISPR-Cas9 model in more detail. Those who are interested may look into that course as well, functional genomics. Yeah. Can you, I mean, the question is that can you have some more practice question for week 4 concept? Week 4 concept, what is the week of concept, Priyanka? Hap map, okay. So we'll see. Certainly we will see that. If you can add more, certainly we'll add. Okay. Continuation with the chromosome integrity. Any known mechanism which holds the telomeric ends chromosome together and in order? Yes, I mean that's the telomere. How they are maintained is sort of well studied using model system, not necessarily from the human cells, but that's now there in majority of the textbook. How, for example, the telomerase helps in because as the cells divide, there's a loss of telomeric length, and there's a telomere enzyme which sort of extend the repeat segments in the telomere to maintain the chromosomal length. So that is pretty much well known, and most of the textbook would offer you, I would say, either the Albert's cell biology or molecular cell biology by Lodish. If you look into, this is available even in the if you go to National Center for Biotechnology Information, NCBI bookshelf, you can type, Google it, NCBI bookshelf, and if you type molecular cell biology or um, um, cell biology by Alberts, this entire chapter is available. You can read that. Um, so there is a please suggest book which will explain molecular biology techniques. Again, there are large number of books. Again, I would recommend you go to the NCBI bookshelf. This NCBI is National Center for Biotechnology Information, and bookshelf. They have a large number of collection of books, very good books in their bookshelf. These are accessible online. You can search using your queries, and you can give molecular genetics, molecular biology techniques. That would really pick up as many books from chapters from that collection. The low dish molecular cell biology also a good starting material even for the molecular techniques. Yeah. Higher prevalence of monogenic disorders could be due to non-allelic recombination. Okay, so that's this is what we have discussed some time back, in the sense that when you have repeats, for example, flanking a gene, um, the same repeat element is present on either side of a gene, and then if there is a um, during recombination in meiosis, what happens if you assume this is my hand, and these are the two homologs, they come to each other, and they come to each other like this because the sequence here and over here are identical. But if I have a repeat and here, and these are identical to repeat that are present in the homologue as well, now the during meiosis, due to some error, the repeats may align like this or align like this. If they align and there is a recombination, meaning there is a double strand break here and there is an exchange, then it would result in, into one of the two homologues having two copies of this gene, or this gene is deleted in the homologue. So that's what happens. So that's what you mean by non-allelic recombination, right? So when they join together like this, and then the recombination, this is allelic. But if they join together in a region, not exactly identical position, but this region there is a repeat, this region there is a repeat, they are identical. If they join, align like this, there is a recombination, that is non-allelic. So that would result in deletion of the intervening segment in one copy and duplication of the intervening segment in the other copy. Can antibiotic resistance be, genetic manipulation? I don't know what is the question is about. I think it's more to do with microbial genetics. Um, well, I mean, if your discussion is about the molecular biology techniques, how do you select the recombinant DNAs? Um, you know, um, I don't know. I mean, certainly this is uh, not something that is related to the human genetics. So it's more of a microbial thing, but that is expected as well, because if you use um, one particular antibiotic for a long time, and certainly the microbes, there will be some genetic change in the genome that would make one or two um, member of a large population to develop a resistance, and if the resistance develops, and they are going to survive, and then your antibiotic becomes irrelevant. And that's the reason why the microbes are living for millions of years, much before we came onto this planet, and they're still successful, so that they undergo changes, and that changes are selected for certain conditions, and antibiotic usage is one. Can we correlate lifestyle habits to integrity and health of chromosomes? Yes, certainly, yes. Um, um, it's pretty well known. The DNA gets damaged, and the damage could happen because of, for example, you are being exposed to UV or X-ray. So these are something that is pretty well known, isn't it? Like, a particular types of skin cancer caused by mutations in the DNA because of UV rays, right? And likewise the X-ray can cause, if someone is working in or living in an area where it is the radiation is very high, or closer to some reactors, or some place where they have stored radioisotopes without the protective shield, there may be leakage and so on. So that is certainly there. And apart from that, there are many other conditions that can cause damages to your DNA. Oxidative stress, people talk about, in your lifestyle, antioxidant. There are, you must have seen so many advertisements. You take this green tea, you take that, and healthy, and so on, which are all antioxidants. So basically, they reduce the level of the ROS, what you call reactive oxygen species, which when the level goes up in the DNA, in the cell, they are going to damage your DNA. So basically, if you reduce the oxidative stress, then you can reduce the damage to the DNA and so on. So certainly the lifestyle is very important for you to sort of make the DNA live longer. Therefore, you can live longer. Could you please explain the mechanism of nonsense mediated decay? Well, there are a large number of enzymes and others involved. Um, that's not so critical here. But what I would say is that um, in a normal, if you look into the human genome, and the genes that are present in a good number of genes in the human have multi-exon, genes. There are multiple exons in a gene, and often the stop codon, that the last triplet in the coding sequence, which gives a signal for stop codon, is normally present in the last exon of the gene. It's in, majority of the genes are like that. Therefore, what happens is when the RNA is being made and the RNA is being transported to the cytoplasm, there is this machinery within the nucleus that pretty much screens the RNA, mRNA, to make sure that that happens when the splicing happens, right? The introns are spliced out. So they pretty much screens the mRNA to make sure that there are no errors in the copying of the mRNA from the gene, and that the stop codon is present in the last exon. Because if there is an error, because anything you mass produce, you're going to have some error, and mRNA is something like a mass production. You have a template, two copies of a gene in an autosomal gene in a cell, and that makes thousands of mRNA. It copies the RNA polymerase copies, and there could be error. And if you allow the an mRNA which has an error, it is not faithfully copied complementary DNA, or whatever is there in the coding sequence. This mRNA with an error in the incorrect base position may end up coding for peptide that are not the correct sequence as per the gene. Therefore, the cell doesn't want to add that kind of a damaged or error prone mRNA to enter the cytoplasm and make protein that are not correct, because that may, one, they may take out a lot of energy out of the system. Second, the mutant protein may affect the cell function. So what the system does is it screens for the stop codon in the mRNA, such that there are no mRNA in which the stop codon in the coding region is present in an exon other than the last exon. If it finds any in which the stop codon is present, in an exon other than the last exon, then it degrades that mRNA. So basically, what happens if you have a mutation, a point mutation, which converts amino acid codon into a stop codon in the first, second, third exon, which is not the last exon, then you pretty much lose the amount of RNA that comes into the cytoplasm. So that's pretty much the answer for brief answer to what you said about nonsense mediated mRNA decay. Where else? What is the? I think that's the pretty much the questions, and I don't see any more questions anywhere. Yeah. Well, if there are no more questions, is there possible? Okay, there are many questions coming up now. Right, so there are, it's possible to have some more video lectures, pedigree pattern inheritance. And certainly not for this run of this course. Maybe in the future it can be done, because all these lectures are recorded some time back. It's not that it goes in live session. A lot of back end work is done, so you can't do it. But you can certainly consider that adding in the next round of offering the course. But in the YouTube, if you go and look at, there are a large number of such video lectures available. Depending on your interest, you can look into. We'll try to add few more practice problems, right? Application of loss of function, gain of function could understood. Okay. Perhaps you are trying to say that you understood now. If that is the case, then we are happy. How to do pedigree analysis of the condition 50% X inactivation? Yeah, so that's going to be extremely difficult. Any conditions in which you have X-linked disorder, it's very, very difficult, because technically there is nothing called a dominant X-linked or recessive X-linked. It doesn't happen because only one of the two X is active in the human female. So what basically you look at is that whether often you'll find the males are more often affected because the 1X, if it contains the mutated gene, then the male would invariably affected. But the male, the transmission from a male always happen to a female, right? And likewise, it always happens from a female to a male because only the female can offer an X chromosome to a male. Um, and of course, if the male offers, then that individual becomes a female, and a male can offer an X to a female, right? So this is how it is because if he offers an Y, then that individual is a male. So these are the other differentiators we look at to see whether it is X-linked, and you are spot on. X inactivation makes it challenging for identifying the pedigree. Okay. Explain between X-linked, recessive, and dominance. That's what I explained now. Right now, theoretically these two are possible, but practically it's very very difficult to really practice because you have only one X that is being active. So there's nothing really much that you can try to differentiate between X-linked, recessive, or dominant. There are some exceptions. There are some regions of the X chromosome that has got genes that have homolog on the Y chromosome. These are very very exceptions. Otherwise, um, I don't see practically is there any difference between X-linked, recessive, or dominant. It's pretty much the same, right? Because only one X is active, and then if the inactive X carries the wild type copy of the X in the female, then that individual develops the disease. But if the inactive X carries the mutated X chromosome, then individual, you know, escape the phenotype. So, but you look at the transmission, whether the transmission is from male to female or female to male, and that's how you sort of tell whether it is X-linked. Good. I think pretty much we have answered most of the questions. If there are more questions, you are welcome to post them on the course portal. We will try to answer them and provide you links for more reading material. I'm sure all of you are finding this course pretty informative and useful. And good luck to all of you for the assignments and the exams. Well, there's some question coming. Krelux system, and this is for the knockout animal. That would be in the fourth. Already we have released that lectures.
Yes,
Yeah,
all released.
I think we have released those videos. These are for making what is called as conditional knockout. For example, you want to knock out a gene in a specific cell type or specific tissue. You basically drive the Krelux deleter, the recombinase in whichever tissue or cell type that you would like the gene to be deleted. So that's what it is. You have a transgenic line in which the recombinase is driven by a promoter that of a gene that is normally expressed in a tissue or a cell type you are interested in. And therefore, it goes and binds to the lock sequence that you have created, integrated in the genome. And then there is what is called as site-specific recombination. So if you have, this is your DNA and you have lock sites here and here, then what happens, the recombinase comes and binds, and then your DNA undergoes this kind of a looping, and then there's a double strand break happens here, and as a result that region is deleted. So that's the principle behind the Krelux system, right? Yeah, I think we have shared with you the reading material. These are from research papers. if you go through them, I'm sure you'll be able to understand. These are very, very easy to follow, the mechanisms. Yeah. I think it's good. I think we should end now because I have another course for which the hangout session should begin in another 5 minutes. So I would like to end here this week's hangout, and you know all the best for all of you for this course, and I'm sure you are enjoying this course. Yeah. Thank you very much. Yeah. Yeah.